Speciation is a process by which new species develop, and it can happen in a number of different ways, but it's really all determined by gene flow. If gene flow between two populations completely ends, then the isolated populations begin to diverge. For example, at one point, South America and Africa were a continuous continent. However, a rift valley emerged, separating the two continents. And over millions of years, the continents separated. And this physical separation isolated the populations of several different species of the two continents, causing new species to emerge. In other words, as gene flow ended, species diverged. But what exactly is a species? It's not as easy as it might seem. There's really three different ways biologists use to define a species, and they all have certain advantages and disadvantages depending on the circumstance. But by biologists, perhaps the most commonly used definition of a species is technically called the biological species concept. According to the biological species concept, a species is defined as a population of organisms that are reproductively isolated from each other. And this can happen in two different ways. In the first way, populations of a species become physically isolated through some natural event and are no longer able to breed due to the distance between them. The divergence of the African and South American continents are a classic example of this. But organisms don't have to necessarily be physically separated in order to not produce viable offspring. For example, what do you get when you mate a horse and a donkey? Well, you get a mule. And if you grew up on a farm, you know that it's impossible to mate two mules to produce viable, fertile offspring. So the biological species concept would say that a horse is a species and a donkey is a species because they can't reproduce and produce viable, fertile offspring. It may be viable in the form of a mule, but mules aren't fertile. So that way, horses are species and donkeys are species, and mules are just hybrids. The biggest advantage of the biological species concept is applicable to many ecological and evolutionary studies. The second major advantage is that species that may be morphologically similar, yet reproductively isolated, are not considered the same species. For example, these are two different species of salamanders based on the biological species concept. Because they don't reproduce even though they look identical and they live in the same area. So if you were in the field, it'd be really difficult to identify these specific salamanders, but in fact they actually are two different species of salamanders because they don't reproduce together. Even though the biological species concept is the most widely adopted definition of a species, it's not used by all biologists due to its specific disadvantages. First, it can't be applied to species that aren't present on Earth right now. So in other words, the amazing resource that we have in the fossil record can't be utilized. And according to the biological species concept, just because a species looks different doesn't mean it is. And in contrast, just because two fossils look the same doesn't mean it's not two different species. Another major drawback of the biological species concept is that it assumes all species have sex, and it's just not true. Many species only reproduce asexually, for example, bacteria. And we know that there's thousands of species of bacteria. And the picture in the middle is a drawing of a flatworm known as a planaria, and it also reproduces asexually. Another disadvantage is really more of a nuance. The biological species concept only applies to populations that geographically overlap. And this is because the biological species concept assumes that populations have the capability to breed, but they don't. Most amateurs use the morphospecies concept to determine what a species is. And if you're a fisherman, bird watcher, or plant collector, you determine a species is based on what it looks like. The morphospecies concept determines that a species is a group of organisms that look, quote, significantly different than another group of organisms. And truthfully, this is how most biologists actually determine what a species is in the field, for the simple fact that who has the time to experimentally mate populations of organisms and raise them to see if they have viable offspring? It's just not feasible at a large scale. And the logic is that structure determines function. In other words, different morphological features arise because The populations are independent of each other due to the isolation of gene flow between them. In other words, structure determines function. And the two biggest advantages of the morphospecies concept are obvious. We can use them to catalog the fossil record. The second big advantage is that we can easily identify species in the field without extensive field work. And also, this concept doesn't philosophically require geographic overlap. So why don't more biologists use the morphospecies concept instead? Well, it's because it doesn't really capture what's exactly going on at the reproductive level. The disadvantages of the morphospecies concept are that it cannot identify species that are not morphologically different. And there's several species in the world that look exactly like one another, but are reproductively isolated. Also, features can be very subjective. And we're now seeing this with the discovery of a hominid species from the island of Florence in the South Pacific. The organisms that these fossils represent only stand about 3 feet tall as adults. The scientists that found these determined that they were new species of humans, Homo florensis. And even more exciting, once they dated these fossils, they were able to determine that Homo florensis lived on Earth the same time as Homo sapiens did. The only other hominid that ever lived on Earth the same time as Homo sapiens was Homo neanderthalis, or the Neanderthals. So this is a pretty significant discovery. However, other hominid specialists have argued that they're really just a subspecies of humans because they share many of the same morphological characteristics as Homo sapiens, despite their short stature. So is it Homo florensis, or just another variation of Homo sapiens? Well, the jury's still out on that one, so keep informed. On the flip side, in some species, there can be enormous variation in physical species. Dogs are a great example of this. Canis domesticus is the Latin name for the common dog species. And they came from wolves, and they include everything from the Chihuahua to the Great Dane. That is some serious variability. But nevertheless, they're all considered one species, even though they're incredibly morphologically different. The new kid on the block is the phylogenetic species concept. With the rapid flood of technological innovations in biology, we can look at huge amounts of data, predominantly DNA. This has allowed us to confirm some of our definitions of species, yet many species have had to be redefined due to this approach. The phylogenetic species concept attempts to define a species not by physical characteristics, but by reconstructing evolutionary histories of populations of organisms. And in this concept, a species is defined as the smallest monophyletic group. So what's a monophyletic group? It's some ancestral group and all of its descendants. It's hypothesized that all life shares a common ancestor. And in this way, all organisms on Earth are in a single monophyletic group. Animals are also in their own monophyletic group. This indicates that all animals also share a common ancestor. However, if you think of life as a tree, species are actually the leaves. The leaf is connected to a branch, which is connected to a larger branch, and so on. Synapomorphies are traits, physical or genetic, that all members of a monophyletic group share. For example, animals are multicellular organisms that ingest food. That's their two major synapomorphies that define an animal: multicellular and ingest food. There is no other organism on Earth that is both multicellular and ingests food that is not an animal. All animals do both of those things. The biggest advantage of the phylogenetic species concept is that it can identify very precise definitions of taxa, even if they look similar. And we don't even need to experimentally mate populations to determine whether they're one or two or even more species. But let me pause for a minute to define the word taxa. Taxa refers to a group of related organisms, and it can be used to refer to all the members of a species, but it could be used to refer to all species in a genus, or even all the species that make up a family, or even crazier, all the species that make up a domain like bacteria. So whenever I say taxa, it just refers to an undefined group of related organisms at some level. One thing that the phylogenetic species concept has been used extensively for in the past 30 years is validating or invalidating previously established taxa. There that word is again. And many species have actually been validated by this process. But when they're invalidated, species are either split into two groups or more, or they're lumped together. Some things we thought were different species, in fact, aren't, and other things that we thought were a single species turned out to be more than one. But the biggest advantage of the phylogenetic species concept is that species are defined based on data, not assumptions. Case in point is the story of the African elephants. For a very long time, it was assumed that there were only two different species of elephants in the world today, African elephants and Asian elephants. However, as biologists began to study elephants more, they began to come across a slightly smaller elephant in Africa with significantly diminished tusk. On further study, it was determined that the smaller African elephants were more or less isolated to the interior of the forest, where they weren't really seen very often, whereas the bigger, longer-tusked elephants were more common to the open plains of the savanna. Biologists began to hypothesize that there were, in fact, two species of African elephants, a forest species and a savanna species. And for a long time, this hypothesis was considered infeasible because of the fact that these elephants exist in the same overall habitat, even though they use different patches within it. These biologists argued that populations would have chance encounters and gene flow would overcome any speciation divergence that would occur. And the data from the DNA sequences suggested otherwise. DNA suggested that there was actually, in fact, two distinct species of elephants in Africa, even though their overall habitat area overlaps. They've differentiated their patch specification to such an extent that they've become completely reproductively isolated, creating two distinct species. Pretty cool. The biggest disadvantage of the phylogenetic species concept is that it leads to the recognition of many, many more species. And many of them look identical. This makes determining a species in the field in the absence of a DNA sequencing machine much, much more difficult. For example, the 16 species you see in this picture used to be one, and I don't know about you, but they all look pretty darn similar to me. The other downfall to this approach is that there are many incomplete phylogenies for many, many different groups. You better believe that butterflies and monkeys are pretty well figured out by now, but who really cares about slime molds and leeches? And without the complete picture, it's really hard to see the forest through the trees. And so once you define a species, it's actually kind of easy to find its subspecies. A subspecies is a group of populations of species that are in discrete geographical areas that have very little gene flow with other subspecies, but they do have some amount of gene flow. And many times, they have very specific traits. So the difference between a subspecies and a species is that a subspecies has little gene flow between different groups, but a species basically has no gene flow between other species. In other words, it's reproductively isolated. Speciation can happen from genetic isolation due to physical isolation. In other words, if two populations get physically separated to such an extent that gene flow declines to zero, the stage is set for divergence to occur. And there are two different ways populations can be physically separated. In dispersal, a group of organisms from one population colonizes a new area. And if there's little to no gene flow with the original population, genetic drift and natural selection can quickly alter the genetic makeup of the colonizing population. This causes divergence, which leads to speciation. In Vicariance, however, a physical barrier splits a population in two, isolating the gene flow from one population to another. And again, the same processes of genetic drift and natural selection cause these isolated populations to diverge, driving speciation. And a great example of Vicariance is the separation of that South American plate from the African plate. As the rift grew larger and larger, organisms could no longer travel across the developing ocean, leading to the divergence of entire communities of organisms. Another example is a lot younger. Even though the Grand Canyon is the largest terrestrial canyon on Earth, geologically speaking, it's actually quite young, at a mere 5 to 6 million years old. You might think 6 million years is a long time, but in reality it's only about 0.1% of the entire age of the Earth. So the Grand Canyon really is very young. Once the canyon began to form, it physically separated a population of squirrels. And over the next 5 million years, the population of squirrels in the North Rim rarely, if ever, encountered the population of squirrels in the South End. And in the 5 million years since, this ancestral population of squirrels have diverged into two different species, the Kaibab squirrel and the Abert squirrel. This is a great example of vicariance. The two forms of physical separation we just talked about are also the mechanism for allopatric speciation. An allopatric speciation is simply when a species diverges into two or more species due to physical separation, and it can either happen by dispersal and colonization or by vicariance. On the other hand, sympatric speciation is the divergence of species into two or more species that live in the same geographic area without geographic isolation. It was originally thought that sympatric speciation was impossible because the gene flow of organisms in the same area would overwhelm genetic drift. But we've actually seen several examples of sympatric speciation in action. One of the possible mechanisms is that different groups of organisms, different populations, have different preferences in habitat, and this leads to divergence. And we've already seen this in the African elephants. We have two different species of elephant that live in the same basic area, but they differ in what habitat they prefer. We have forest elephants and we have savanna elephants, and they don't really merge. In this way, competition for resources are limited because these species are eating different food sources. In other words, these elephants have different preferences in their habitat, forest elephants and savanna elephants. Here's another example of sympatric speciation in action. These two different fly species live in the same area and diverge from the same ancestral species, and they look kind of similar. However, they consume very different resources. Apple flies pollinate apple trees, whereas hawthorn flies pollinate hawthorn bushes. Duh. In this way, they're not competing for the same resource even though they live in the same habitat. Polyploidy is a chromosome-level mutation in which the entire karyotype of an organism is duplicated. In other words, it leads to individuals having more than one set of chromosomes. And it happens during mitosis. What happens is that mitosis continues on like normal through anaphase, but telekinesis doesn't proceed, leaving an extra set of chromosomes in the cell. For example, when a diploid or 2N cell duplicates but telekinesis doesn't proceed, a 2N cell becomes a 4N cell. We call a 4N cell a tetraploid. So in the example on this slide, we have a cell that has 3 chromosomes, so its 2N equals 6. But when the diploid replicates, it becomes a tetraploid, and therefore 4N equals 12. This can also happen during meiosis as well. Polyploidy is relatively rare but not totally absent in animals. Typically it produces either non-viable or non-fertile offspring. However, it's really rare in plants, and it can cause rapid speciation simply for the fact that organisms with different numbers of chromosomes can't reproduce and produce viable offspring. Let's see an example. Seedless watermelons are a miracle of modern science, even though they've removed children's abilities across the world to have seed spitting contests in hot summers. But you may have wondered, if they don't have seeds, how did they come to be? Good question. Seedless watermelons are actually a hybrid of two watermelon strains. One is a diploid and the other is tetraploid. During meiosis, the diploid produces 1 N gametes and the tetraploid produces 2 N gametes. When you add 1 and 2 together, you get 3, right? The zygote, in the case of the watermelon as a seed, is triploid. In other words, you have 3 chromosomes. Now this grows into a perfectly functional watermelon, except it can't grow seeds. Why, you might ask? Well, when a triploid cell goes through meiosis, all of its gametes have an uneven number of chromosomes. And since they're uneven, they're not able to produce viable offspring. In other words, they can't produce seeds. Brilliant. But wait, you might be thinking, all right, well, it's fine to produce that one watermelon, but how do you support an entire industry of seedless watermelons? Well, there's two possibilities. The first, you can take clippings off a watermelon plant that's seedless and asexually propagate many of them to produce new plants, or you can simply repeat the original procedure of mating diploid watermelons with tetroid watermelons. And each works. Okay, so explain this. How do you get square watermelons? Well, the answer is you grow them in boxes. That's really what they did. This is from Japan. And the story behind these are Japanese people love watermelons, but they have really limited shelf space. So what do? They make square watermelons. Crazy! All right, let's bring it back in here. There are two forms of polyploidy, autopolyploidy and allopolyploidy. Auto is Greek for the same, and autopolyploidy is when members of the same species produce a zygote from two diploid gametes instead of two haploid gametes, producing a tetraploid zygote. And this has happened in maidenhair ferns. Two diploid gametes have fused, creating a tetraploid zygote. And since tetraploids can't produce viable offspring with diploids, the two groups have been genetically isolated from each other, causing divergence. And eventually this causes sympatric speciation. While autopolyploidy is when polyploidization happens with members of the same species, allopolyploidy happens when chromosomes merge from different species. And when this happens in plants, we get hybrids. These are usually tetraploids that come out of the fusion of diploid gametes, and sometimes they can be viable and other times they can't. Horticulturists have produced several variants of flowering plants using this method of hybridization. The main reason we see polyploidy in plants in greater abundances than in animals is because many plants have the ability to self-fertilize. And when this happens, with a plant that has a mutation that creates diploid gametes, those diploid gametes can fuse, producing viable offspring that are able to reproduce with each other. And those offspring are tetraploid. Another reason that plants are more commonly polyploidies than animals is that they have a greater ability to hybridize, creating another opportunity for allopolyploidization. So what happens when species that have diverged geographically come back together? Well, of course the answer is it depends. But it really depends on how far the species have diverged genetically. And if there's been a large and significant divergence, then mating is extremely rare and probably non-existent. And even when those populations do come back together, their gene flow is not really happening at all. And those populations continue to diverge. However, if the divergence is insignificant, the populations are able and willing to mate with greater frequencies. Gene flow increases between the two disparate populations, and the populations converge back together. So what happens when isolated species come into contact that actually have the ability to interbreed? They create what is known as a hybrid zone. And this is a geographic area where the inbreeding of two species occurs. The most common result of the hybrid zone is that the hybrids that are produced are evolutionarily inferior to either of the pure species, and the hybrid typically becomes locally extinct. However, every once in a while, hybrids are actually more suited to their environment and able to outcompete the original species, and this can actually lead to the origin of a new species.